So thanks very much, Matt, and it's a real pleasure to be here today and catch up with people that we know and work with. And I want to give you an update on where we're up to with ophthalmic genomic medicine. So basically, this is my overview slide to indicate that really this is a partnership between what's happening for the people affected with the conditions. Whoops, that's not, sorry, Emma, next one. Oh, yeah, right, good. So what's happening for the people with the conditions, the genomics and the genetics work that we can do related to that, and then all of the cell biology work that we can do and the animal model work to work towards the novel therapies for these conditions and other ways that we have to work with them. So thinking about it from the patient and affected people perspective, so there's a lot of input, as a lot of you would know, from paediatricians, from ophthalmologists, from clinical genetics, and we're also working all the time then towards the genomic aspects, particularly when there are conditions with a genetic cause. And you've probably heard about the various revolutionary things that have happened in the genome sequencing world, and that's really made a huge difference to people with these conditions in terms of getting a genetic diagnosis and then working towards improved therapies in these conditions. And we work towards those in lots of ways, using the cell studies that I talked about. I'll talk about the human-induced pluripotent stem cell studies that are also very useful, and also, of course, mouse and other models. And this gives inheritance information for families and also helps us work towards gene replacement or other potential genetic therapies or pharmacological therapies or whatever is appropriate for the particular circumstance. And as John indicated, we also have a health economic study because, of course, we have realised that interacting with the various boffins that we definitely need to have definite health economic data and social impact data that can go with that to indicate that all of the needs that are in these areas. Next, yep, okay. So I'll just talk about two main areas, so developmental eye disorders and the retinal diseases. So the developmental eye disorders are things that affect particularly often can be the front part of the eye there that you can see, the cornea, the iris, the lens. So conditions where there's congenital cataract, where there's severe clouding of the cornea. And these conditions, like the retinal conditions, can be caused by many, many different disease genes. And so that's been an enormous problem in terms of trying to get genetic diagnosis for people. And as you can see by the diagram there on the right, there's a lot of overlap between the gene that can cause an anterior segment abnormality, a gene that can cause a cataract problem, and so on. So it's been very difficult up until the advent of next generation sequencing to actually being able to get a genetic diagnosis for people. But we have used, in the research that we have been doing, we have been using next generation sequencing, so whole exome, whole genome sequencing, and we can now get a genetic diagnosis, 70% mutation detection rate for cases where there's congenital cataract. For anterior segment disorders like Peters anomaly, you can get a 50% mutation detection rate. That's not published yet. We're still working on that paper to get that out. And so for 2/3 of the families, that's usually given new information about the diagnosis and the inheritance pattern for that family. The problem used to be that we used to have to try and look at one gene at a time. And you saw as that previous slide, there were over 100 genes that you had to look at. And so with the technology that we used to have, so we could only look at one gene at a time, which each of those genes, it would take about 6 months to look at them, and it would cost about $2,000. So if you had to look at 100 genes, $2,000 times 100, it just wasn't possible. And in fact, the testing wasn't available. The game changer that's been is this thing called next generation sequencing, whole genome sequencing, whole exome sequencing. And so with that, you can actually look at the whole genome at once, and then you can select. What we do is we then select the particular eye genes that we want to look at, and that's how we can get a 70% mutation detection rate for people, because there's many possible causes of a particular phenotype. You can't just look at one gene and expect to find the answer. You've got to look at at about 200 of them to find that answer. So we're now able to do this as a clinical diagnostic test. So Children's Hospital Westmead Laboratory does that for us. And if you feel that would be helpful for you and your family, then any clinical genetics service can refer for this testing. Now, of course, there's a 70% mutation detection rate or 50% mutation detection rate. That means that there's 30% or 50% of people where we're not finding answers. So then that is a subject of further research studies. And we're particularly using particular whole genome sequencing approaches in those cases to try and get to the bottom of what is causing the problem. And there's various types of genetic variants and so on that come into play there. So there is still research to do in this area, even though we have already made a lot of progress, but there's still research that we're working on in that area. So similar concepts apply for the genetic retinal diseases, and there are other genetic diseases, of course, that affect the eye, including corneal diseases and so on, but these are just my two example cases at the moment. And in these conditions, again, there's marked phenotypic and genetic heterogeneity, and so there's a lot of overlap in those genes. There's over 200 disease genes. So it's been very tricky. Most people, a lot of people, haven't had a genetic diagnosis for many, many, many years. And these conditions affect about 1 in 3,000 people. So this is not uncommon disease. And so this is really making a difference. And again, we have clinical diagnostic testing now implemented for those conditions. So again, any clinical genetic service will be able to order that testing. And again, we're getting this sort of 50 to 70% detection rate, and we've still got further research to do to help find the answers where we don't currently know them. But what this has opened up is we're able to find what's called new genotype-phenotype correlations. So, for instance, this gene was only previously known to be associated, caused Leber congenital amaurosis. But what we found through this testing is that it could be a cause of cone or cone rod dystrophy. So that's important for us in refining those testing strategies that we have. We now run a multidisciplinary team meeting where we meet with the laboratory people. We have clinical genetics people there as well, and we have the ophthalmology teams as well, because it can be tricky sometimes to sort out, is that variant really causing the condition, or is that just a common variant? We've all got variants in our genes. That's why we're all a little bit different in our own way. And so it's sometimes a little bit tricky to sort that out. So we do need a bit of work. It still takes time to sort through this, but we are getting those detection rates that I mentioned. So what this has done is opened up a whole new world in this area, because once we can get a genetic answer, then we can work towards potentials for genetic therapies for conditions where previously there was no possible option in these cases. And so one of the ways that we're working towards this is by making human-induced pluripotent stem cells and working with human-induced pluripotent stem cells. So that's where you take a piece of someone's skin and actually reprogram the cells so that they can then turn into any possible type of cell. And of course, what we do is turn them into eye cells. And that is a great way. Oh, sorry. Yep. Thank you. That's a great way to model, to work towards therapies or improve strategies for management for these conditions, because the problem with the eye is that a lot of the genes are only expressed and doing their work specifically in the eye. And so it's hard to study because, so you can imagine a muscle disease, you can actually do a muscle biopsy and take a piece of someone's muscle and do all your laboratory work that you need to do on that piece of muscle. The problem with the eye is tricky. We can't take a piece of someone's retina and do all our cell biology and experiments that we need to do on that. So therefore we have to develop these model systems. We have relied on animal models up until this point in a lot of cases, but it's a fantastic technological breakthrough again to be able to take that piece of skin and turn it into retinal pigment epithelium, which I'm showing in this particular case. So the retinal pigment is at the very back of the retina and provides nutrients and support for the photoreceptor cells. And these are cells that are very important and are in fact the disease cells in some cases of the retinal dystrophies. So they form this very nice cobblestone appearance there. So this is from a person with a particular eye problem, and we can then study their retinal pigment epithelial cells in the dish. And that is a very powerful way then of, if you want to test like a new pharmacological drug therapy, or if you want to test a genetic therapy, which I'll go on and talk about, or gene replacement therapy. So the other thing that we do is you can differentiate those same cells into what's called a retinal organoid. So you can see there on the day 30 slide, that's a little developing retina. So all the retinal layers that are in that we normally have, so the photoreceptor layer cells and so on, we can see them developing in those retinal organoids. So again, that's a powerful way of testing a potential therapy. And this is just a picture here. So the photoreceptors have these little connecting cilium, you can see it there, and they're actually developing in those retinal organoids. Next one. Yeah. So one of the things that we do is called genome engineering, and you might have read about this on social media, I'm not sure. So it's CRISPR-Cas9 gene and base pair editing. So what we've had to do for the genetic therapies mostly until this point of time have only been for those cases where we might be able to do a gene replacement. So in that situation, the mutation is causing loss of function of the whole gene, and so what we do is we're adding extra gene for the genetic therapy. Now, the trick is that doesn't work for all cases, and in some cases you actually have to work hard to correct the particular base pair that's causing the problem. And so this CRISPR technology is able to actually cut the DNA and then get the cell to correct. You can add in the correct base pair in that spot, and you can actually correct the DNA in that spot. And so what we're working towards, what it would be great, would be able to do that in the eye cells, so that we could do a correction in the actual eye cells themselves, and so that then the cell could start functioning, the photoreceptor could start functioning properly, and so the generation wouldn't keep happening. So these, of course, are very research phase at the moment. So the gene replacement is actually in clinical trials and FDA approved for RPE65, as John said, but the gene and base pair editing is still very much in the research phase. So the other work that we are working on is new approaches in delivery of the therapy. So once we're working on the gene replacement constructs, we're working on the base pair editing approach. What we need to do then is be able to get those constructs which are doing the therapy into the right place at the right time. And there's a little virus that's used to do that. It's sort of like a cold virus, but it's even more benign than that. And what we do is we manufacture that so that it's not pathogenic in any kind of way. It's called adeno-associated virus, so that's AAV up there. And what we're working on is improving the capacity for those viruses to get to the right cells at the right time. And we're using those retinal organoids and the other cellular approaches that I talked about to do that. So all of this work, of course, does takes time, and so we've also just luckily received a New South Wales OHMR grant to work on a clinical grade vector production facility. So because, of course, if we're working towards these sorts of therapies in the human eye, we have to have clinical grade production of the vector, which is a big deal in itself. And so we're part of a grant that's going to be working towards that. And the very exciting thing is that the eye is at the forefront of this, because for this vector production for other genetic diseases, like that it might affect the liver, other parts of the body, you actually need huge amounts of this vector, which is a very expensive exercise. Whereas the eye, it's a smaller organ, so you need less of it. So it's seen as a great starting place for therapies in this particular area. And then there's also, of course, regenerative medicine approaches where you can actually do cell transplantation. Of course, that's very much in the research phase still. And I'll just show you the pictures here. So we also work with the mouse to do testing in the mouse, and that's a little mouse over there having their vision tested. They also sorry, Emma, if you just go back. Yeah, sorry. They also get OCTs, the mice, and they also get electroretinography. So they get the full gamut. They get the full gamut. But that's really important to test these new therapies because, of course, the government, we're not going to be able to administer these towards patients without, adequate testing and so on. So just next slide. Yes, thanks. So this is it's a big production to really work towards these sorts of areas, and we have to be very mindful of all of the areas that we're working on. We have to have the real clinical expertise. We have to have the proper ophthalmic diagnoses to understand what we're dealing with. We have to have the genetic clinics working fully, the laboratory work happening so that we can get the genetic diagnoses adequately. We have to have the multidisciplinary team meetings, as I said, are very critical, and we're now developing this health economics arm, which again is critical to, I think, be up there convincing government that this is all very important for our patients. We've got, I've mentioned the GMP production and, of course, then working towards the clinical trials in these conditions. Next one. So I just want to thank my research lab, who are very dedicated people who work incredibly hard. It's a very complex and detailed molecular biology, cell biology to work all of this up, and they work incredibly hard. So I want to thank them, and I want to thank you, and I'd be very happy to talk later. I'm sure we're probably running out of time, but very happy to talk later about any questions that you might have. Thanks.
Thank you, Professor.